I thought since today's theme is all about sample preparation, I wanted to talk a little bit about how we can use CryoSEM for imaging challenging biological samples that could be difficult or impossible just using conventional SEM. So not to confuse it with FibSEM, I'm going to be talking today about just standard CryoSEM. So maybe I'll step back and talk a little bit about the research that I did before at USDA, where I spent basically a decade running the EM facility there. And this was a really high throughput lab. So we were receiving, samples on a daily basis from around the world, and we needed a technique that would be capable of handling the diverse range of samples and the kind of high throughput that we had. And so CryoSEM really turned out to be a really great technique for that. So maybe I'll explain a little bit about like why you would want to do CryoSEM. Of course, everyone's familiar with the kind of challenges that you're facing when you're trying to do fixation and dehydration and critical point drying. Some of these samples are very difficult to, properly prepare without getting them shriveled and really a lot of distortions. And in particular, these mites that I was working with were really, really small, like 300 microns. So even if we did dry them, they'd be very difficult to manipulate and kind of transfer between all of those vessels. And then when they're dry, they're like statically charged. So what we found is that, CryoSEM turned out to be a much better technique for us. And so this is what we were able to achieve through CryoSEM. And you can see this is a mite. This is a citrus leprosis or mite that causes citrus leprosis disease in citrus plants. It's really prevalent in Mexico, and it's kind of inching closer to the US border and threatening our citrus industry. Basically, it carries a plant virus, and it transfers those viruses to plant to plant as it feeds. So in this image, you can see that the mite is very, it's in a lifelike state. It's not squished down onto carbon tape or glue. It doesn't have glue all over it. It's actually on the actual leaf itself. So you're capturing actual realistic behavior of the mite on its native host there, the plant. And if you look closely, you can actually see it's actively feeding. It has its kind of stylet inside the plant. And so this was really important for us to understand, because we were trying to figure out how this virus is getting transferred, and we could see that it's actually, from the mite feeding on the plant. So this is kind of one technique that, really wouldn't have been possible through conventional SEM. So maybe I can give some other examples. I did a lot of research on mites, but also looked at a wide range of samples. So this is an image of some bacterial spores, and this is kind of a string of spores that it's just kind of extending out into free space. So if you were to now put your electron beam on that sample, you'd start to see a lot of drifting and stuff. And so CryoSEM, it helps to control the drift as well. And so that worked out really well for that sample. This is another sample of some algae that was really nicely imaged. This is an example of a bacterial colony growing on some stainless steel metal. So this is really important for food safety researchers who are trying to understand how bacteria are growing on different surfaces and how they can, when you're dealing with vegetable processing plants, how do you keep those surfaces clean? And what we found is that different bacteria like to grow in different ways, and some like to stay on top of the metal, and you can see the metal grain boundaries in the background. And other bacteria like to, kind of go into those little cracks and crevices and are much harder to sanitize. And so we were experimenting with different coatings and trying to find antimicrobial coatings. And so you can imagine if you were trying to do this with conventional SEM, as soon as you put this into fixative and all these different reagents, a lot of those bacteria would probably most likely get washed off. And so with this method, we were able to just freeze it instantly. This is, of course, just a nematode, a plant parasitic nematode. And one thing that you can notice about it is it's very kind of plump, and it almost looks muscular. This is maybe a little bit different than how you might have seen SEM images of nematodes in the past, where it looks like more like an earthworm with all the segments. A lot of the morphology of different insects and mites has been described based on actual chemical artifacts that that change the shape of the organism, and so we're actually describing species based on artifacts, chemical artifacts. So that's really not the direction that we want to pursue as biologists. We want to be, studying these things based on realistic characters, right? This is another example of a plant leaf surface. So you can see all the trichomes very nicely preserved. Yeah, no dehydration or shriveling there. And then again, you have another example of, in this case, an aphid on a leaf, and you can see it's in a very natural kind of posture. Its antenna and its legs are all kind of in a lifelike state. So, if you're a researcher who's studying these insects now, you have a realistic image and not a specimen that's been smushed down onto carbon tape. Another great thing you can do with CryoSEM is of course, freeze fracturing. When you fracture things under cryo conditions, the surface breaks along these natural fracture lines, and so you can get images like this. So this is a leaf where you can see palisade, mesophyll, spongy mesophyll, and some vascular area. In this case, we were studying beech leaf disease, which is kind of spreading in the East Coast here and damaging a lot of our beech trees. And so we got some samples from some researchers, and it turned out that when we fractured this open, we saw all these nematodes inside. So colorize the nematodes. Again, that's helpful for researchers and also people who are reading the papers to understand what they're looking at. So this is now was published as a causative agent of this beech leaf disease. This is another cool example that I spent a good portion of my time working on at USDA. So we were studying colony collapse disorder in honeybees, and this is a major problem throughout the world. Our honeybee colonies are getting diminished by this mite called the varroa mite that feeds on bees and transfers a couple different viruses. But for the last 40 years or so, everyone was under the impression that the varroa mite only feeds on the larval stage of the bees that are inside the actual honeycomb. And the thought was that they're only using the bee to transport and to move around and kind of like a taxi. But we noticed something strange. This mite was always located in a very particular spot here underneath these tergites of the mite, of the bee. And so there was something going on there. Why does it have this preferential spot? And so we wanted to see what was going on underneath there. So we took this image. This is a SEM image, even though it's been colorized, of the whole bee. And we were able to get a closer up mag of the mite on the bee, with its head is actually tucked underneath these tergites. Yeah, so we wanted to see what was going on underneath there. And so we tried this a couple times. We imaged it, and then we pulled the sample out, and then we actually used some tweezers, and while it was still partially frozen, we removed the bee, and we got an image that looked like this. And so this is the first time we had ever seen something like this. This is looking kind of at the soft membrane of the bee, and we can see there's this impression that is left behind in the soft membrane of the mite. And you can see the individual segments. Of course, I've colorized this, You can see the hairs, and in fact, the feet of the mite were really well attached. So when we pulled the body off, those feet stayed attached. So that actually gave us additional confirmation that yes, this is an actual the feeding or the location where the mite was, and you can see it lines up perfectly with what the mite actually looks like. But there's something else going on right here in the center. It looks like some kind of a mound or something. So let's take a closer look at that. And we see, okay, we see a hole now. So this appears to be a feeding wound, right? And so this is the first time that anyone has seen this. And we can see there's two of these really distinctive grooves here, kind of a larger one and a smaller one. And that actually, fortunately, lines up perfectly with the mouth parts of the mite. We have these two, the chelicera and I think the fixed digit there. So yes, this is basically conclusive evidence now that the varroa mite is actively feeding on adult honeybees, and this is kind of overturning about 40 years of kind of false belief that they didn't feed. And we followed up this work with some fluorescence work to track the food as it went into the mite, and some TEM cross sections as well. And so this was published in 2020 in PNAS, and a lot of cool microscopy in that paper. So if you want to take a look, feel free. So it's just a couple examples of how you could use CryoSEM, some of the advantages that it might provide. Maybe I'll talk really briefly.
Hey, Joe?
Yep.
Before you go too far, what was the significance of that exact location of where that mite was attacking?
Yeah. It's because of there's some fat bodies cells that are underneath that location. And so that's, yeah, there's actually food underneath there in that specific location that it can feed on. Yep.
Is it on both sides of the bee?
It is on both sides. Yeah, so we do find them on both sides, but for some reason there's a little bit of preference on one side. So, but yeah, we did some TEM work as well to really show the fat bodies and right underneath the surface. So,
got it. Thank you.
Yep.
Since we are pausing, Emily, you have your hand raised. Do you have a question to ask? Okay, I guess not.
Okay.
All right. Sorry, Joe. Keep going.
So yeah, I'll talk a little bit about the hardware. So we're using the Quorum system. This is the Quorum PP3010T. This is a really nice kind of compact cryo system. And so there's essentially three main steps here, right? You mount your samples and you freeze them. You have a slushing container here. And then you can touchscreen monitor where you can control all your temperatures, and a lot of this is basically fully automated, so you just set it and go. After you freeze your samples, you transfer them into the cryo prep chamber. Here you can do a couple different things. You have two different knives where you can do freeze fracturing. You have a small scalpel that's really for fine movement, and then a heavy duty kind of steel microtome blade. And then we have integrated sputter coater. So you can do a platinum sputter coating, for example. You can also do sublimation to kind of remove any residual frost that's on the sample. So a really nice compact system. After you do your coating, you can then transfer that into the SEM chamber, and you have a cold stage that's fed by cold nitrogen gas. So this is, you can run this at, somewhere between -40 and -60 typically for up to 24 hours, and do a lot of imaging, and your sample stays cold, and it helps you to image without having any kind of beam damage, right? Because your sample's frozen. So the nice thing about Zeiss and pairing this system with a Zeiss SEM, it can go on any of our SEMs, essentially anything from our basic Evo all the way up to our very technically complex crossbeam. We have workflows and for every system here. I'm actually most excited about our Sigma 360. This came out last year as our kind of updated system for core facilities. This is at the really kind of the right balance of resolution and price, and also kind of being a robust system that can be great for a core facility where you have a lot of users. It's not going to break down. It's really a workhorse SEM. And the good thing about it is it has this Gemini column, which is specially designed and tuned for really great low kV imaging. So it's very common for us to image at 1 kV or up to 500 volts, for example. And that's basically due to a lot of the optics, and I can tell you about that at a later time. But so why would we want to image at low kV, right? This is an image of some macrophage shells. So you can see in the left here, this image is very nicely detailed. There's a lot of surface detail. And that's because the electron beam is traveling at a low energy, only 1 kilovolt. And so the electrons are just interacting with that surface and then generating your image based on that. If you now increase your electron beam to, like, say 10 or 15, now your electron beam's penetrating a lot deeper and you're getting a lot more subsurface imaging or detail that's coming through, and which is typically not what you want for biological samples when you're trying to image, morphological details, right? So low kV is always going to be better. And of course, if you now pair the CryoSEM with an SEM that can image out very nicely at low kVs, then you can get some nice results like this. This is a hydrogel sample from Nina at North Carolina State University, and we were able to image these samples without any coating at all. So yeah, this is a protein kind of fibrogel, and we have a leaf vasculature that's been freeze fractured. Neither one of these have been coated. So there may be certain applications when, based on the research or the biology, that you don't want to coat something. And with this technique, with CryoSEM combined with low kV imaging, you can do that. Of course, coating always gives you better conductivity, so it's good to coat your samples when you can, but if you, for some reason, you can't, you have that option with the Gemini column. So I think that's my time. I'll leave it there. This is a short tech talk, but I guess I'll give you one more shout out to my BioEM team. We are a newly formed team here at Zeiss. We're specializing in life science research and BioEM. And so my specialty and area is the East Coast here. If you're on the West Coast, you can please feel free to reach out to Steven Hernandez. We also have our Aubrey Funk as our product marketing manager, and and Philip Bastian as our North America Life Science sales manager. So any questions or any kind of information that you guys have, we're here to help. We have a lot of BioEM experience between all of us, and we're very happy to share information, techniques, and yeah, please feel free to reach out.